Alright, let's go ahead and get started. Physiology and histology was the one. Oh, yeah, and then we had 2 hours of clinical pathology. It's no wonder I'm starting to stutter and not speak right. Just been talking already for long enough. Okay. So, when the osmolarity is the same in something, whatever you're measuring, things aren't going to move. Okay? So, no water is going to move if the area is isotonic. Outside of cells, sodium is abundant. And we're going to go ahead and talk about this chart here. So, in hyponatremia, the cause is usually adrenal insufficiency or the overuse of diuretics. The plasma sodium concentration is going to be decreased, extracellular fluid volume is going to be decreased, intracellular fluid volume is going to be increased. Hyponatremia and dehydration was that one. Next is hyponatremia due to overhydration or hyponatremia paired with overhydration, caused by excess ADH or bronchogenic tumors. It's going to result in a decreased plasma sodium concentration, an increased extracellular fluid volume, and an increased intracellular fluid volume. Hey, young Fayez, how you doing? Now, hypernatremia paired with dehydration can be caused by diabetes insipidus or excessive sweating. It results in an increased plasma sodium concentration, a decreased extracellular fluid volume, and a decreased intracellular fluid volume. Hypernatremia and overhydration can be caused by Cushing's disease or primary aldosteronism. Results in an increased plasma sodium concentration, an increased extracellular fluid volume, and a decreased intracellular fluid volume. Okay. Hyponatremia basically just means less sodium outside of the cells, where it's supposed to be. So, let's talk about edema formation. Edema is excess fluid in body tissue. Most cases of edema are going to be in the extracellular fluid compartment. We talked a little bit about this a couple days ago, but this is more in depth. It can also occur in the intracellular fluid compartment as well. So there's pitting edema, which is extracellular edema. It is due to abnormal leakage of fluid from plasma to the interstitial space and failure of the lymphatics to return blood from the interstitium back into the blood. Can also be due to excess capillary fluid filtration. Some factors that increase capillary filtration are Starling forces. So, it's a graphic. I don't know how to describe it really. Okay. Causes of extracellular edema. Here's some more. Lymphatic blockage is one, and then filarial nematodes, heartworms, or cancer. Those can all be causes. Heart failure can be a cause. Increased venous and capillary pressure with increased capillary filtration. Decreased arterial pressure, which means decreased excretion of salt and water by the kidneys, which leads to an increased blood volume and capillary hydrostatic pressure. And then increased renin, angiotensin II, and aldosterone secretion, which leads to more salt and water retention. This is how heart failure can cause edema. So, can also happen due to decreased kidney excretion of salt and water, which that can happen due to acute glomerulonephritis, which is failure to filtrate adequate amounts of fluid. Okay, and can also happen due to decreased plasma proteins, which decreased plasma proteins can happen due to a failure of the body to produce normal amounts of proteins, or liver cirrhosis, or leakage of proteins from the plasma, or nephrotic syndrome, which is damage to the glomeruli. Now, intracellular edema, which is non-pitting, like if you were to stick your finger in it, there wouldn't be a pit when you take your finger out, right? That's what non-pitting edema is. This happens due to sodium pump depression, which is basically where, increases in intracellular sodium osmolarity can cause water to move to the inside of the cell. Okay, and it can also happen due to inflammation, which is an increased, or leads to an increased permeability to sodium and other ions, leading to osmosis of the water. Most edema, again, is extracellular, and it's rarely intracellular. But there's factors in the body that prevent edema, otherwise we'd just be walking around with edema, right? Low compliance of the tissues in the negative pressure range is one. Lymphatic capacity for increasing flow is two. And the washdown of interstitial protein is the third one. And edema happens when those safety mechanisms are no longer there, once the body's failing. So talking about that first one a little bit more, low compliance of tissues in the negative pressure range. Hydrostatic pressure in most subcutaneous tissues is around -3 millimeters of mercury. This is the negative pressure range, so the compliance is going to be low. But when it goes above 0 millimeters of mercury, the compliance becomes high and the safety against edema is lost. So, next is the lymphatic capacity for increasing flow. Basically, the lymphatic system returns the fluids and proteins to the circulation and takes it out, etc. Without that function, there's going to be a decreased plasma volume and edema occurs as a result. Lymph flow increases 10 to 50 fold, which means 10 to 50 times normal when the fluid starts to accumulate, and that can lead to edema. Not good. Finally, washdown of interstitial protein. An increased fluid filtration, an increased pressure, and an increased lymph flow, and a decreased protein concentration, a decreased colloid osmotic pressure. All of these lead to the net result being decreased net filtration across capillaries. Not good. Okay. Moving on. Potential spaces. So we've talked a little bit about potential spaces before. That's like your peritoneum, your pleural spaces, right? These are spaces that aren't spaces because they're normally empty and they have a negative pressure, but they could be a space. That's why they're called potential spaces. Like if you were to get stabbed or something, not good, but that's how it could be a space. Plural: peritoneal, pericardial, synovial. What happens is fluid accumulates because of unbalance or imbalance of capillary forces or lymphatic blockage due to infection or injury. And then effusion is where edema happens in the potential space. Okay, moving on to glomerular filtration and renal blood flow. We kind of went over the anatomy of the kidney already. Went over that. So there's the Bowman's capsule and the glomerulus. Very important. In the first segment of the nephron is where most filtrate is reabsorbed. That's the proximal tubule and the loop of Henle. Okay? The nephron is the functional unit of the kidney, though. Good to keep in mind. You cannot get nephrons back. They're like brain and heart cells. They do not regenerate. So take care of yourself. Take care of your kidneys. Kidney innervation. Kidneys are innervated by sympathetic fibers only. This is the smooth muscle of the renal artery, right? Interlobar, arcuate, and interlobular arteries. Afferent and efferent arterioles, renin-producing cells, proximal tubules and distal tubules, the loop of Henle, and then the collecting ducts, all innervated by sympathetic fibers only. There's no parasympathetic innervation in the kidney. Now talking about micturition, peeing, right? Larger animals have more volume for pee. But interestingly enough, it seems all mammals pretty much pee at the same, not the same rate, but takes them the same amount of time to empty their bladder. About 20 seconds, 20 to 25. When the bladder's full, stretch receptors send signals to the brain, right? And then there's a signal that goes back to the detrusor muscle and to the internal urethral sphincter, and that is what causes you to get that tingling sensation like, oh, I need to go pee, right? But as you get older, starting at infancy, you're unable to control when you pee. You just, whenever you get the urge, you just pee. You just make a mess in your diaper, right? But when you get older, you get control of something called the external urethral sphincter. This is a voluntary sphincter, surrounded by voluntary muscles, which allows you to hold your pee. Okay? As time passes, however, the forces and urges can get stronger until you just pee yourself because you can't hold it in anymore, because your bladder is going to rupture if you do. So it's probably a good idea to use a restroom before that point. Now talking about the anatomy of micturition or urination or peeing. You've got the bladder, the ureter or ureter, external sphincter, right? All these, and then there's the nerves. So sympathetic, parasympathetic, and pudendal. It's different in males and females, obviously, different organs. Peristaltic action along the ureters is initiated by the stretch of the renal calyces as urine passes out of the collecting ducts. Peristalsis is enhanced by parasympathetic and inhibited by sympathetic stimulation. But in the bladder, it's a two-step process. the filling of the bladder and an increased wall tension. And then there's a reflex. There's a nervous reflex called the micturition reflex that empties the bladder or induces the desire to urinate. And then there's a reflex from the autonomic nervous system from the spinal cord to your bladder and all that stuff. These can also be inhibited or facilitated by the cerebral cortex or the brainstem. So, again, as the pressure increases in the bladder, you just get an increased urge to urinate. It's common sense, really. I mean, everyone experiences it. As the bladder fills, sensory stretch receptors send signals via the pelvic nerves to sacral segments of the spinal cord near your pelvis, right? That's the lower spine. Parasympathetic stimulation of the bladder's smooth muscle via the same pelvic nerves occurs. It is self-regenerative, the urge to urinate. It'll come back, and then it'll subside, and then it'll come back. It happens over and over and over and over and over until you're able to urinate, until your external sphincter is relaxed and you're peeing. So, but it's facilitated and inhibited by the brain, right? You can say, I don't want to pee right now because I don't want to pee my pants, or there's no toilet around, okay? Or you can say, I do want to pee right now because I'm in the woods, or there's a toilet and I'd like to use it. But you yourself you can control whether or not you pee or don't pee. I mean, it's common sense, but this is what we went over in physiology and histology. Blah, blah, blah. The steps in the involuntary urination. Okay, this is kind of just common sense. Your abdominal muscles contract, which increases the pressure in the bladder. Extra urine enters the bladder neck and posterior urethra, which leads to wall stretching. And then stretch receptors are activated, which leads to the micturition reflex and inhibition of the external urethral sphincter. There are some abnormalities associated with micturition or urination. An atonic bladder, that's where the sensory fibers are destroyed and overflow incontinence occurs, which that happens when people get syphilis, urinary incontinence. There's an automatic bladder, which happens when there's a spinal cord injury above the sacral region. The micturition reflex is still intact, but it's uncontrolled, and it leads to frequent urination. Then finally, there's uninhibited neurogenic bladder. This is a loss of higher center inhibition. This leads to interruption of inhibitory signals, which leads to frequent urination. Not good. They're all pathologies. Or they're all disease processes. Anyway. Okay, that was it for physiology and histology. For that day, at least. Okay, moving on to clinical pathology. Oh, the 15th, I think, right? Yeah, 15th. Well, I'm going to go ahead actually and end this video, take a small break, and I'll be back. But thank you all for watching. I'll see you in a bit.